The envelope sat untouched on the polished mahogany desk for 3 days. Its contents would eventually reshape how humanity understood itself, yet the man who penned its questions had grown accustomed to being dismissed. In 1875, Francis Galton was obsessed with a question most scientists considered unanswerable. What makes us who we are? Is it blood or is it circumstance? He believed twins held the key. The scientific establishment thought him eccentric at best, delusional at worst. But Galton would not relent. He sent hundreds of questionnaires across England, meticulously collecting stories of twins who looked alike, twins who did not, twins raised together, twins separated by fate. What he uncovered would ignite a debate that burns to this day, and buried within his own data lay something he never anticipated, evidence that the very heredity he championed had limits he could not explain. If you find this story fascinating, take a moment now to subscribe to this channel. The history of human genetics is filled with brilliant minds, dark chapters, and revelations that change everything we think we know about ourselves. More stories like this are coming. Francis Galton entered the world on the 16th of February, 1822, in Birmingham, England, into a family of exceptional intellectual pedigree. His mother, Violetta Darwin Galton, was the daughter of the legendary physician and poet Erasmus Darwin. That connection meant young Francis shared a grandfather with someone who would eventually change the course of scientific history, Charles Darwin. The two boys were half-cousins, linked by blood and by a family tradition of relentless curiosity. But while Charles would spend decades studying finches and barnacles, Francis took a different path. He was a polymath in the truest sense. Geography, meteorology, statistics, psychology, anthropology. No discipline was safe from his probing intellect. He invented fingerprinting as a means of identification. He pioneered weather maps. He developed statistical concepts still used today. And he harbored a conviction that human traits, from height to intelligence to moral character, were passed down through generations like heirlooms. Galton's childhood revealed a mind that could not be contained by conventional education. He learned to read before the age of 3. By 5, he could recite Latin poetry. His sister Adele, 12 years his senior, took special interest in nurturing his gifts. But the formal schooling he received left him cold. He detested the classical and religious instruction that dominated English academies of the era. In a letter to his cousin Charles, years later, he confessed that traditional biblical arguments had made him feel wretched. He craved empirical truth, measurable data, the cold clarity of numbers. Medicine initially seemed a suitable outlet for his intellect. He studied at King's College London and later read mathematics at Trinity College, Cambridge. But his heart was never fully in the healing arts. Then came the inheritance. When his father died, Francis Galton found himself wealthy enough to abandon any profession he did not love. He threw himself into exploration and adventure. In 1850, he organized an expedition to Southwest Africa, traversing regions that few Europeans had mapped. He documented the flora, fauna, and peoples he encountered with the precision of a trained naturalist. When he returned to England, he published accounts of his journeys that earned him recognition from the Royal Geographical Society. But travel was merely a prelude. The real work of his life was still waiting. Everything changed in 1859. That was the year Charles Darwin published On the Origin of Species. The book detonated across the intellectual landscape of Victorian England. Evolution by natural selection, the transmutation of species, survival of the fittest. These ideas rewrote the rules of biology. For Galton, they did something even more profound. They gave scientific legitimacy to questions he had been asking since boyhood. If species changed over time through the inheritance of favorable traits, then surely the same mechanisms operated in human beings. Surely genius, talent, and virtue passed from parent to child, just like the shape of a nose or the color of an eye. He began devouring Darwin's work, corresponding with his famous cousin, pushing the ideas further than Charles himself was willing to go. Galton's first major work on the subject arrived in 1869 with a book titled Hereditary Genius. In its pages, he argued that exceptional ability ran in families not because of upbringing or opportunity, but because of inherited biological material. He studied the genealogies of judges, poets, scientists, and military commanders. He traced lines of brilliance across generations. Critics objected. They pointed out that the sons of famous men also enjoyed superior education, social connections, and resources. Galton anticipated this objection. He needed a way to separate the effects of biology from the effects of environment. He needed a natural experiment that would isolate heredity from everything else. And so he turned his attention to twins. The logic was elegant. Twins represented the closest thing nature offered to a controlled experiment in human development. If two individuals shared the same blood, the same parents, the same home, then any differences between them could only be attributed to some factor beyond heredity. Conversely, if twins who started life alike remained alike despite different experiences, that would prove the supremacy of biology over circumstance. Galton set about gathering evidence with characteristic thoroughness. He designed a questionnaire containing 13 groups of questions. He sent copies to twins themselves and to their relatives. The final question asked recipients to provide addresses of other twins who might participate, creating a self-expanding network of respondents. He received replies from 80 sets of twins, though he focused his analysis on 35 cases that provided the most detailed information. What emerged from those questionnaires both confirmed and complicated Galton's expectations. The twins who looked alike at birth remained alike into adulthood with remarkable consistency. Their hair color, eye color, height, weight, strength, and vocal patterns matched so closely that even family members struggled to tell them apart. Some correspondents described twins who suffered identical illnesses at identical times, despite living in different locations. Others reported twins who arrived at the same ideas, made the same decisions, and pursued the same interests without any contact. One respondent described twin brothers who independently purchased identical gifts for their wives on the same day. Another told of twin sisters who fell ill with the same fever within hours of each other, though separated by hundreds of miles. Galton collected these anecdotes with delight. They seemed to prove that nature stamped its template upon humanity with indelible ink. But the data contained a more troubling pattern that Galton addressed only briefly. Among the 20 cases of twins who were dissimilar at birth, not a single pair grew more alike, despite being raised in identical circumstances. This finding cut both ways. It suggested that no amount of shared nurture could overcome an initial difference inscribed by nature. But it also revealed something Galton seemed reluctant to confront. If even a shared environment could not make different twins alike, then heredity imposed limits on what it could transmit. The machinery of inheritance did not produce perfect copies, it produced variation, and that variation could not always be explained by the laws Galton was trying to formulate. This is a good moment to pause the story and ask for your support. If you are enjoying this deep dive into the origins of genetic science, please hit the like button. Your engagement helps this channel continue producing content like this. Now, back to Victorian England and a man whose obsession was about to collide with the limits of his own theory. Galton published his findings in 1875 in an article titled The History of Twins, as a criterion of the relative powers of nature and nurture. The title alone announced his ambition. He was not merely studying twins. He was using them as a criterion, a measuring rod, to weigh the fundamental forces shaping human destiny. He introduced the phrase nature and nurture, a formulation that would echo across centuries of scientific debate. He concluded that nature prevailed enormously over nurture. Similar twins remained similar regardless of circumstance. Dissimilar twins remained dissimilar regardless of shared upbringing. Biology was destiny. Environment was a minor actor at best. The scientific community received Galton's work with a mixture of curiosity and skepticism. Some researchers recognized the originality of his method. Others questioned whether anecdotal questionnaires could substitute for rigorous experimentation. Still others worried about the implications. If human traits were fixed by heredity, what did that mean for education, social reform, or moral responsibility? Galton himself seemed untroubled by such concerns. He pressed forward, refining his ideas, gathering more data, constructing ever more elaborate statistical tools to analyze the patterns he observed. In 1883, he coined a new term for the application of these principles to human improvement: eugenics. The word derived from Greek roots meaning good birth or well-born. Galton envisioned a society that encouraged the most capable individuals to reproduce while discouraging or preventing reproduction among the less capable. He believed he was founding a new religion of human betterment grounded in scientific fact. The twin study of 1875 became foundational to this project. It was cited by researchers around the world as the first systematic attempt to quantify the relative contributions of heredity and environment. Edward Thorndike, an American psychologist, conducted tests on 50 sets of twins in 1905 and reached conclusions similar to Galton's. Hermann Werner Siemens, a German dermatologist, expanded the twin method in the 1920s, using it to investigate the genetic basis of skin diseases. Each successive study built on the framework Galton established, yet each also encountered the same anomalies that lurked in Galton's original data. Twins were not always as alike as they should have been. Environment seemed to matter more than the theory allowed, and the boundary between nature and nurture proved far more porous than anyone expected. The shadow of eugenics grew darker as the 20th century progressed. In the United States, advocates of better breeding pushed for laws restricting immigration, segregating the unfit, and forcibly sterilizing those deemed genetically defective. By 1937, more than 30 states had enacted sterilization statutes. The Supreme Court upheld these practices in a chilling ruling that declared 3 generations of imbeciles are enough. Across the Atlantic, the ideology took an even more horrifying turn. Nazi Germany embraced eugenic thinking with murderous enthusiasm. Josef Mengele, the physician known as the Angel of Death, established a gruesome laboratory at Auschwitz, where he subjected twin prisoners to unspeakable experiments. He injected chloroform into the hearts of children. He sewed living twins together to create artificial conjoined pairs. He dissected bodies searching for genetic secrets. 1,500 twin pairs passed through his hands. Most did not survive. The connection between Galton's questionnaires and Mengele's atrocities was not direct, but it was unmistakable. The logic of hereditary supremacy, taken to its extreme, produced horrors that stained the science of twins for generations. After the war, twin studies continued, but under a pall of suspicion. Researchers sought to distance themselves from the Nazi taint while still pursuing questions about heredity and environment. The most influential figure in this post-war era was a British psychologist named Cyril Burt. Burt claimed to have conducted extensive studies of identical twins who had been separated at birth and raised in different homes. This design represented the gold standard for disentangling nature and nurture. If twins with identical genes grew up in unrelated families yet still resembled each other, that would demonstrate the power of heredity more conclusively than any previous research. Burt reported just such findings. He put the correlation between the intelligence scores of separated identical twins at 0.771. In paper after paper, he presented data showing that genes determined intellectual ability regardless of upbringing. His work influenced British educational policy, including the introduction of the 11+ examination that sorted children into academic and vocational tracks, based in part on IQ tests. But Bert's empire was built on sand. Shortly after his death in 1971, his records and notes were destroyed by fire. Researchers trying to verify his findings could not locate two of the three assistants whose names appeared as co-authors on his papers. When asked about them, Bert said they had emigrated, but he did not know where. The third collaborator appeared to be entirely fictitious. Statisticians noticed suspicious regularities in Bert's published correlations. In one paper, he reported testing 21 twin pairs, in another, 53. Yet the correlation coefficient remained identical to 3 decimal places, 0.771. The probability of this occurring by chance was vanishingly small. In 1974, the American psychologist Leon Kamin exposed these anomalies. Bert's approved biographer, after extensive investigation, concluded that his twin studies were fabricated from beginning to end. The scandal destroyed Bert's reputation and cast doubt on the entire enterprise of separated twin research. Into this breach stepped Thomas Bouchard, an American psychologist based at the University of Minnesota. In 1979, Bouchard learned of two identical twin brothers who had been separated at birth and reunited after nearly 4 decades apart. James Springer and James Lewis had grown up 40 miles apart in Ohio, each unaware the other existed. The adoption agencies had told each family that the other twin had died. When they finally met, the similarities astonished everyone who heard the story. Both had worked as deputy sheriffs. Both had done stints at McDonald's restaurants and gas stations. Both drove light blue Chevrolets. Both had dogs named Toy. Both had married women named Linda, divorced them, and remarried women named Betty. Both had named their first sons James Allen, differing only in the spelling of the middle name. Both suffered migraines that began at age 18. Both gained 10 pounds in their early 30s. Both chewed their nails and chain-smoked Salem cigarettes. The Jim twins, as they became known, launched a media sensation that carried them from newspaper front pages to The Tonight Show starring Johnny Carson. Bouchard seized the opportunity. He invited the Jim twins to Minnesota for extensive testing. He issued public appeals for other separated twins to come forward. Over the following years, his center accumulated 81 pairs of identical twins and 56 pairs of fraternal twins who had been raised apart. The results, Bouchard reported, confirmed the hereditarian hypothesis. He estimated that adult intelligence was 70% heritable, meaning that 70% of the variation in IQ within a population could be attributed to genetic differences. The findings reinforced a worldview in which biology trumped environment. Destiny resided in the genes, and social interventions could do little to overcome the hand dealt by nature. But Bouchard's work attracted criticism from the start. His twins were self-selected volunteers who had already found each other and spent time together before entering the study. Those whose lives most resembled each other were most likely to seek publicity and contact researchers. The sample was not random. It was biased toward similarity. Moreover, nearly all the twins in the study had been raised in white middle-class American environments that were far more alike than the methodology assumed. The premise that separated twins experienced entirely different environments simply did not hold. Adoptive families, as one critic observed, were all alike in the ways that mattered most: stable, aspirational, adequately resourced. When you controlled for this shared background, the apparent power of heredity shrank considerably. The Jim twins themselves, examined more closely, revealed differences that the sensational coverage obscured. When they first met, One sported an Elvis-style haircut, while the other had a Beatles look. One was talkative and outgoing. The other was reserved and preferred to write down his thoughts. Springer stayed with his second wife, Betty. Lewis married a third time. Their children had different names and were born at different intervals. The uncanny coincidences that made headlines, the Salems, the Chevrolets, the dogs named Toy, were products of chance amplified by selective reporting. Among nearly 2,000 twins studied over the decades, bizarre parallels would inevitably appear, but they represented statistical noise, not evidence of genetic determination. The deeper problem lay in what twin studies could not measure. Identical twins share not just genes, but also the prenatal environment. They usually share a single placenta. They experience the same hormonal fluctuations, the same nutritional inputs, the same stresses during gestation. Any effects traceable to these shared conditions would appear as genetic influence, even though they were environmental in origin. After birth, identical twins continue to experience correlated environments. They look alike, so people treat them alike. They often dress similarly, attend the same schools, join the same social groups. Every experience that reinforces their similarity gets counted in the heredity column, even when it belongs elsewhere. Fraternal twins, by contrast, diverge from each other both genetically and environmentally. One may be larger in the womb and crowd out the other. They each have separate placentas. Their different appearances invite different treatment from parents, teachers, and peers. Any study comparing identical and fraternal twins will tend to overestimate genetic effects because it cannot fully account for the divergent environments fraternal twins experience. Consider a practical example. A pair of identical twin girls are born with slightly above-average cognitive abilities inscribed in their genes. Because they look alike, their parents dress them alike, enroll them in the same classes, expose them to the same books and activities. Their teachers, unable to tell them apart, give them identical feedback. Their friends overlap. Their experiences reinforce each other at every turn. By adolescence, their IQ scores are nearly identical. Is this because their genes determined their intelligence, or because their genes triggered a cascade of environmental responses that amplified their initial similarity? The twin study cannot answer this question. It can only observe the correlation and attribute it to heredity. Now consider a pair of fraternal twins, one a boy and one a girl. From birth, they are treated differently. The boy is encouraged toward sports and mechanical toys. The girl is steered toward dolls and domestic activities. They make different friends. They develop different interests. They read different books. By adolescence, their cognitive profiles diverge. Is this because their genes destined them for different paths, or because their different treatment from infancy onward shaped them into different people? Again, the twin study cannot distinguish. It sees divergence and calls it genetic. James Flynn, one of the most influential IQ researchers of the past half-century, articulated this problem with characteristic precision. He observed that identical twins born with sharper-than-average brains would independently seek out similar environments. Both would go to the library. Both would gravitate toward advanced classes. Both would attract the attention of teachers who nurtured their abilities. Their genes did not directly produce their adult intelligence. Rather, their genes created a multiplier effect by prompting them to seek experiences that enhanced their initial endowment. The environment was not a passive backdrop. It was an active participant, shaped by the very traits it was supposed to explain. To credit heredity alone was to mistake the seed for the entire garden. The limits of heredity became even clearer in the 21st century as new technologies allowed researchers to study genes directly rather than inferring their effects from twin comparisons. The sequencing of the human genome in 2003 delivered a profound surprise. Scientists had expected to find around 100,000 genes. They found only about 20,000, roughly the same number as a roundworm. This meant that the complex traits studied by psychologists and social scientists could not be controlled by single genes operating in isolation. Intelligence, personality, mental health, and behavior emerged from intricate networks involving thousands of genes, each contributing a tiny effect that could be amplified or suppressed by environmental factors. Genome-wide association studies allowed researchers to calculate polygenic scores that captured the combined influence of many genetic variants on a particular trait. When these methods were applied to educational attainment, the results were sobering. A study of nearly 55,000 Icelanders found that genetics accounted for only 17% of the variation in how long people stayed in school. This was a far cry from the 50% or higher estimates produced by twin studies. The gap suggested that twin research had been systematically overestimating heritability by conflating genetic and environmental influences that traveled together. Epigenetics added another layer of complexity. Scientists discovered that environmental pressures could alter gene expression without changing the underlying DNA sequence. Stress, nutrition, trauma, and exposure to toxins could switch genes on or off, producing effects that sometimes persisted for multiple generations. This meant that the boundary between nature and nurture was not a fixed line, but a dynamic interface. Genes responded to environments. Environments triggered genes. The dichotomy that Galton had enshrined in his phrase nature and nurture turned out to be a false binary. Perhaps the most striking evidence of environmental influence came from studies of twins raised in genuinely different circumstances. Bouchard himself acknowledged that some of his separated identical twins showed IQ differences of 24 or even 29 points. This was a staggering gap, larger than the average difference between random strangers. If genes determined intelligence, how could two people with identical genomes differ so dramatically? The answer had to lie in their different experiences. In 2014, a remarkable case emerged from Colombia. Two pairs of identical twins had been mixed up at birth in a hospital error. One twin from each pair was sent home with the wrong family. They grew up as fraternal twins, unaware that their true genetic matches were living in a different city with a different family. When the error was discovered decades later, researchers tested all four men. The results confirmed what the hereditarian model struggled to explain. The twins who had grown up in the same environment were more similar than the twins who shared identical genes but different upbringings. Rural poverty and urban middle-class life had left their marks in ways that genes could not override. A similar pattern emerged from Korean twins separated by adoption. One was raised in Korea, the other in the United States. Despite identical genomes, their personalities, values, and cognitive styles reflected their different cultures. The American twin had absorbed the individualism and optimism of her adopted society. The Korean twin displayed the collectivism and emotional restraint of her homeland. Heredity had provided a starting point. Environment had written the rest of the story. Galton died on January 17, 1911, at the age of 88. He had lived long enough to see eugenics embraced by governments and intellectuals across the Western world. He believed he had discovered the principles that would guide humanity toward perfection, but history would judge his legacy harshly. The forced sterilizations, the immigration restrictions, the concentration camps, all these horrors drew intellectual sustenance from the conviction that heredity determined human worth. Modern genetics has dismantled the scientific foundations of that conviction. We now know that genes do not operate as simple blueprints. We know that environment shapes their expression at every stage of life. We know that the traits Galton measured, intelligence, creativity, moral character, emerge from interactions so complex that no formula can predict them. Yet Galton's twin study remains a milestone in the history of science. It was the first systematic attempt to use natural experiments to separate hereditary and environmental influences. It established a methodology that researchers refined for over a century, and within its own data, it contained the seeds of its eventual correction. Those dissimilar twins who never grew alike despite identical upbringing, those similar twins whose minor differences hinted at environmental influence, those anecdotes of illness and coincidence that could not be neatly sorted into genetic or experiential categories. Galton's obsession led him to nature's door, but the evidence he gathered revealed that the door opened onto a landscape far more complex than he imagined. The story of twin studies is not over. In 2025, researchers at King's College London published the largest genome-wide association study of identical twins ever conducted. They examined data from nearly 22,000 identical twins drawn from 11 studies across multiple countries. Their goal was not to prove that genes control behavior, but to understand how genes modulate sensitivity to the environment. The findings were subtle and striking. They identified genetic variants associated with greater responsiveness to environmental influences. These variants did not determine outcomes directly. Instead, they shaped how strongly individuals reacted to the experiences life threw at them. Some people carried genes that made them more vulnerable to trauma, but also more responsive to supportive relationships. Others carried genes that buffered them against adversity, but also dampened their response to enrichment. The old question of nature versus nurture gave way to a new understanding. Nature through nurture, genes, and environment intertwined in a dance that neither partner leads alone. As we conclude this exploration, take a moment to consider what Francis Galton could never have known. He lived before the discovery of DNA. He worked before anyone understood chromosomes or gene expression or epigenetic modification. He asked the right questions with the wrong tools and reached conclusions that later evidence would complicate and partly overturn. But his determination to measure what others dismissed as unmeasurable opened a path that science still walks today. The twins he studied, the questionnaires he sent, the data he gathered, all of it contributed to a conversation that continues in laboratories and universities around the world. If you found this journey through the history of heredity enlightening, please subscribe to this channel and share this video with others who appreciate deep dives into science and history. The story of how we became who we are is far from finished, and there are many more chapters to explore together. The envelope that sat untouched on Galton's desk in 1875 contained replies from twins whose names are now forgotten. But their testimony lives on in every genetic study, every psychological assessment, every debate about what makes us human. Galton believed he was proving the supremacy of nature over nurture. Instead, he helped reveal the intricate partnership between them. His data showed that twins raised alike remained alike, but it also showed that twins raised differently became different despite sharing every gene. He thought he was closing a question. In reality, he was opening an inquiry that grows more fascinating with every passing year. The limits of heredity are not a defeat for science. They are an invitation to deeper understanding. Every person is shaped by genes inherited from ancestors stretching back through millennia. But every person is also shaped by the womb that nurtured them, the home that raised them, the schools that taught them, the friends that challenged them, the culture that surrounded them, the historical moment that defined their possibilities. Francis Galton's obsession with twins led him to a truth he could only partly grasp. We are neither prisoners of our biology nor blank slates written entirely by experience. We are the products of both, woven together so tightly that no instrument can fully separate them. That is the legacy of his twin studies. That is what the data revealed when examined with modern eyes. And that is why the conversation he started continues, a century and more after his death, in every laboratory and every family where the question is asked, what makes us who we are?